get some water, but in my own cup. Thank you. Oh God, I don't want to start laughing. Like, that.
I know we're gonna get the giggles at some. It's not gonna be good.
Someone did a great job on
We're not professional at all.
whoever was their
About that.
Yes, we knew that about us. They don't.
We still have some street cred left.
Yes, we're not anymore. Enjoy the next 10 minutes.
Now, what's your all's relationship with FAU? I know that FAU has a bunch of
After that,
To be fair, we have not been on time for any session all day, so I wouldn't worry about it.
why us? Oh God.
That's good though.
Yeah, we're gonna need the wine bar after this.
Although myself in one goes like this. I don't think that Guinness doing my Bible study.
When did you have a Guinness? Jenny!
Hey, hi. Nice to see you in person. Yeah.
Let's go.
We're stressed at home. I mean, that's just me probably.
No, I think that's probably everybody.
But it's not because of any.
No, it's us.
Coordination was. I have had all day to ramp myself up.
To like peak levels of. Yeah, I know.
Yeah. No, that's how we do it.
Well, we've been.
We're not gonna let you down.
Okay, because like I said, I see their education materials pop up when I go
Okay, thank you so much.
for various things. Just right up with the FAU and places.
I don't want to be a member of any club that was happening.
We don't remember. That's
Yeah.
like Groucho Marx.
Yeah. I do not want to be a member of any club that will happen.
That's me, the story of my life.
Essentially.
You like me, oh, there must be something wrong. Like, you try to find the fault of something. That's amazing.
Yeah, there must be something wrong with you. Or me, or both. Or,
Both, yes.
yeah.
There's so much more that's wrong.
It's gonna be fabulous. People are gonna be fainting.
It's gonna be fresh.
Like the Beatles, they'll be screaming, throwing things at us.
That's probably.
That may not be a good thing.
That's a truth that you threw out there.
So I know that they're presenting
How
many pets did you get?
So many. But out of all the ones that I managed to get, I think my dreams are special. Like this guy.
thing, and then there's a little bit of
It's one of the minions, not minions, in Toy Story.
kind of excited.
I send my husband that one at least once a week. He'll say something completely banal, and I'll be like,
Not a buyer, but a war buyer.
it does look fancy. I like the one with the little flashlight. Oh, is it gel? Maybe is that from IDT?
Yeah.
Oh, I gotta go get one.
We need to go back. Yeah, we need to go back and revisit some.
And grab the pipe pens and yeah.
We have places to go. Because I love chalk pens. This is my weakness.
Khalil and I were pen swabs, and like when Lorraine came, she's like, "Oh, we're like, do you want any special pens or anything?" And she's like, "Oh, I don't care." And we're like, blasphemy.
It's like it's not sparkly and like purple and like everything, like gel and like the fine lines and thickness. And this is like Japanese company that makes this stuff, but those are like again like you have to
But like Japanese stationery is like, oh,
oh my god.
I am such a jet pens junkie.
Just like again, favorite brothers together.
I gotta show you the notebook I bought for. Oh, right, we'll be like Japanese stationery 101.
And I'm like, okay.
Yeah.
I am passionate about that stuff. And then my husband's like, how many pens do you need? Like, mind your own business.
There's no limit.
There's no matter.
And when you find a
I
good
don't know
one,
how many pens I need, but I know how many husbands I get.
And that's zero.
True.
And then when you find a really good one, you have to get it.
All the colors.
Yeah, oh yeah, because if it dries really well, you want it in all the colors, like, yeah. All right, 26, 4 minutes, countdown, final countdown. Okay. Fourth door,
Yeah,
426,
Kate, really.
426.
What's going? There's no clock in here. It's like Vegas.
It's 426, we have 4 minutes.
We have 4 minutes.
Phone's off. Yes.
Phone's on silent. Oh, we should check into our own session.
Oh yeah.
Oh my God.
No, this is like, we're amazing.
We're amazing. It's gonna be great. I don't know how my name ended up at the. Did they put us in alphabetical order? I feel bad.
Yeah, I think it's actually that's true. You have a strawberry hair, like
I'm too
You
old to
win.
be cute.
you.
I'm too old to be cute.
Went
It
over, you
goes.
think?
Well, no, it goes over. Like it goes in. It's like a session and a half or something.
Oh, it's okay. And we're not responsible for that.
It's all good.
2 minutes. We're starting on the dot, I guess.
Yeah, I suppose. People can trickle in.
But that's what I was saying. We've not been on time for anything. I don't know that we should start this dot.
Maybe
Because ourselves would miss the beginning.
We
Yeah.
would miss the beginning. This is true.
by waiting now. So 1 minute in, 2, let's say 4:32. Executive decision.
There you go. It's all good.
I'm gonna start talking about
Talk about
Oh
ideas.
my goodness. Yeah, don't know. Just no.
that could have gone badly.
That'll be fun. I use a different word that's way worse. See, you knew what I meant.
I knew exactly what you meant.
But I reserve that for special occasions. It's not like you don't throw it around because like, wait.
No, you had that's gravitas. Yes.
Yeah, very fancy of you.
Your SAT word of the day is.
Yeah. The word that I was thinking or the word that you were thinking?
I don't know.
There's so many things we can like combine.
We could. I just remember taking the GRE and like doing the English part and going, are any of these words actually in English?
No, the Greek, most of them are Greek.
Yeah, Greek or Latin.
I aced the GRE like I was like, I don't know, 1 percentile or something because
Because everything was in Greek. Yeah, I'm like, I
these are
didn't go
just
to
regular
Philip
words,
Exeter.
y'all.
No, yeah. This is a piece of cake. I'm sitting there going, what in the Phillips Exeter is this crap? None of this is in English.
which was actually true.
Yeah, see, there you go.
That's what I tell my kids if you want to go to medical school.
Study Greek.
Study Greek. Yeah. Learn Greek so you can talk to grandpa and go to medical school.
Oh, I'd have to show you. Coffee stain.
Oh.
See?
Dude.
We're matchies. Coffee stain.
Twinsies.
I can't take you guys anywhere.
Twinsies. Hey. 2 minutes more and we start.
All right.
I think what I will do at the beginning of the recording is I will call out people that I know that should come to my session and did not.
I think they're already recording, so you could just go ahead and do it. I hope they trim these things. Ooh.
Yeah, let's get started.
Do you need to do anything?
introduce the LMRG.
Ah, yay, that's right. Okay,
See, I know what's going on, y'all.
because you were paying attention,
Was it though?
unlike the rest of us.
I'm supposed to. I will. Just give me 1 second. Hello. Hi. Hi. Thank you for coming to the Lightsheet Technologies Realities for Incorporating Lightsheet into a Core Facility. To start off, though, we're going to have a presentation from Erica from the LMRG, and she's going to tell us about the LMRG.
Hi, my name is Erica Wee. I'm the co-chair of the Light Microscopy Research Group, and thank you for your just 1 minute pitch about the research group. It's a community of many of you here are very passionate about imaging and image analysis, and we also work with broader community like BINA and Global Bioimaging for standards and rigor and reproducibility. And we have been doing ongoing bioimaging community discussions, the topics from image analysis, image segmentation, AI in bioimaging, and we'll continue this trend, while including more topics for 2026 to 2027. And just a quick plug that tomorrow morning we have a talk from 10:45 on statistical considerations for microscopy data interpretation, and also a AI bioimaging analysis workshop. And if you would like to join us, please scan the QR code or reach out to me. My name is Erica, and you can find me through the ABR app. Thank you. Thank you, Kay.
Thank you, Erica. Oh no. Y'all, I lost the cursor. What? What?
Oh, did it become a pointer?
Aha. I still don't have a cursor. Come on.
Oh, it's down. I saw it.
I think it's. Oh, okay. All right, y'all. Just kidding. That's right. I can run a microscope. Thank you for coming. My name is Kay Thomas, and today we have a panel. We're going to talk a little bit about our light sheet technologies that we've incorporated into our core. Today I have with me Christina Labno. She is the director of the Integrated Light Microscopy Core at the University of Chicago, where she received her PhD. And Christina has been at the core there for about 20 years, and she became the director of the core, facility director, in 2023. Christina Piergocki is the head of the Advanced Microscopy and Innovation Lab at Memorial Sloan Kettering Cancer Center, and she's been there since 2024. She spent 8 years at the Rockefeller University Bioimaging Resource Center, about half of which where she served as the operations manager. She's going to tell us a little bit about her journey, but also just to hit the highlights, she was a products and application specialist at Zeiss and received her PhD from the University of Colorado Health Sciences Center. So, sorry. Okay, so without any more drama, okay, so I'm Kay. I'm going to start us off. The goal here is to give you kind of an overview of what our facilities are and our kind of perspectives, and then open it up for a discussion for any questions that you might have or what you might be considering if you're considering incorporating Lightsheet into your core, or maybe some advice that we can have that's helpful. I am the technical director of the Center for Advanced Light Microscopy at the University of Rochester Medical Center in Rochester, New York. And a little bit about my facility. So we're a team of 4. We have an umbrella structure of the Center for Advanced Research Technologies, and there's 9 cores that kind of fall under that structure. The main thing I want to just point out is that we have 200 users that use our core from about 100 different labs. So you can see that means that some of our labs may be giving us 1 user or 2 users And so there's it's kind of an interesting mix of our users. We have 800 instruments, and we book about 7,000 hours a year. We operate on some institutional support as well as a chargeback model. And then our equipment is funded by a mixture of external grants and institutional funding. So these are our microscopes. So we have a range of confocals, we have super resolution, we have multiphoton, we have our light sheet, and then we also offer like a microscope, an epifluorescence in a box, and as well as a couple of workstations. So that's kind of the breadth that we're working with. We have everything from the basic systems all the way up to expert line STED. And so there's a kind of a whole range of what we're working with here. So some of you may know what light sheet is. Just to kind of orient everybody, we're just going to give a quick brief overview but not go too far into it. So light microscopy, the basic thing that you have to remember about light microscopy is that you're going to be using an orthogonal pathway, an orthogonal light pathway. This allows you to kind of have the advantages of wide field detection with a rapid, gentle imaging, but very fast for larger volumes. And so this is one of the images that we took on our light sheet. There are lots of different ways to light sheet, and there are different ways to think about it, so I'm just going to strip it down to the very basics. So when you think about traditional microscopy, you have a light source. The light source is sending the light into the sample, and then that same light source is generally collecting the light from the sample for fluorescence microscopy. If you rotate this on your side, you still have that one objective sort of system flowing. What you're going to do with light sheet microscopy is you're going to throw in another objective or light source into the system that's going to kind of cut across your sample. And then you're still going to have your detection objective that's going to collect the data. And so you can have more than one of those illumination objectives. And then what you usually do is you focus on the light sheet, and then you move your sample through. So you can see like how the sample moves through, and you get like these slices sections of your sample. Now, there are many ways to set up this type of system. You can have the two objectives coming from the top, from the bottom, from the side. You can have one objective with a second objective that's hidden behind it. Like, there's all sorts of things that you can do. The system that we have is one of these with the four objectives that comes together. So the multi-view. So we installed our movie SPIM, was installed in 2023. We basically have all three octagons, as well as six laser line system. It has 2 separate cameras, and this is kind of some of the specs for our system. What's really interesting about this system is that we have 3 different octagons, so it's basically kind of 3 systems in one. So the 20X system has the 4, so 2 illumination, 2 detection objectives. It's a dual-sided for faster imaging. You can also rotate the sample if you need to, and there's also the possibility of environmental control. Some of the things, though, is that this has a very small area for imaging inside, and so you're really limited into your sample size. And so you'll see down here in the corner, I did have a pointer, but I don't know what to do with it. You'll see down here in the corner some of the specs for like what you can put in that chamber. So some of the things that we've had success imaging in that is like Drosophila and zebrafish embryos, organoids. We've even done some protein polymerization assays, things that don't work that people often think of when they think of like live light sheet imaging as cultured cells, traditionally mounted samples on slides, or even large cleared samples. This holder's not compatible with solvents. So there are some, and the processing is complex because you have 2 different images that you kind of have to overlay with each other. So then we also have the cleared octagon. So this one has 2 illumination objectives and a single detection objective. It's compatible with organic cleared solvents, and it can take pretty high resolution images. We've actually been able to see like dendritic spines on this instrument. And this is for the mid-size to lower end of the larger samples. Things like cleared mouse brain, like half a mouse brain, embryonic mouse heart, lungs, organoids. We've been able to do that. Generally, a whole brain doesn't fit very well into this container, and limbs like hind limbs and things that aren't cleared do not work well. It also struggles kind of with the more jelly cleared types of samples because of the way that it holds the sample. You get like some recoil as it moves through, so it does work better with like the iDisco stiffer samples. The alignment is difficult. And the chamber's not really easy to kind of cycle between different solvents. So to kind of supplement that, we also have the 4X, which allows us to image larger samples like a whole brain, like a hind limb, or like a foot. And you can see here is one of our hind limb images from one of our researchers. But again, we're still limited. You're not going to be able to put a whole mouse in there or anything that's very, very large. And it doesn't really work very well with anything that's very, very small. It's slightly easier to clean this one because the objectives don't dip it the same way. So we use this one kind of as our mounting media flexible kind of chamber. So it's a little bit easier to transfer it between different medias. The alignment is a little bit more forgiving because of the lower resolution. So overall, we just have the Bruker movie spin. And we had all three instruments. And so the one person, so me and another scientist in the lab, we're the ones that work with the system the most. And I was like, what would you say about the system? And we swear at the system all the time because it's difficult. But she was like, honestly, it's a pretty good system. We get really high image quality. We have great response time from our company partners. The system is really flexible, so we are able to image everything from live samples all the way up to very large cleared samples, which for a smaller core like us, we're only going to have one instrument. We're not going to have 3 different specialized instruments. And so this does allow us to kind of take all comers and give that technology to all of our users without kind of having to just be like, okay, we can only do this one thing. It is very customizable, which also means that it's a lot of work to align it, but the alignment is customizable. We have a lot of degrees of freedom for like kind of adjusting things. There's a very steep learning curve in the mounting and alignment, processing. All of that is different than anything that most of us have ever done before, especially when you're bringing users into the system. We struggle with the chromatic shift correction and the color dispersion. So I'm hoping to get some tips today about that. And switching between octagons is actually is not trivial. So I was talking again to my scientist and she's like, it's as hard as switching between different instruments. So if you think about using like 2 different types of confocals or something like that, it's just as hard to make that switch as it is to switch between like our live cell octagons and our cleared cell octagons. So there's a very big difference there between how you would kind of just think about the workflow and how everything works. And then, like I said, even though you think you could use everything on here, we kind of are limited to kind of one type of media at a time. So we can't just switch between, disco and clarity as easily as you might think. And with that, I'm going to hand it over to Christine. Oh, I forgot to say, and we'll do questions at the end.
Oh, yeah, we decided we're going to do all the questions and discussions at the end. All right. Oh. And I'm already hitting the wrong buttons. All right. So as Kay said, I'm Christine Labno. I'm the director of the Integrated Light Microscopy Facility at the University of Chicago. We were founded in 1997, so the Core is going to have their 30th anniversary next year, which kind of freaks me out a little bit. We are a centralized core, so we serve the entire University of Chicago community, but most of our users come from biological sciences, chemistry, and the Pritzker Schools of Medicine and Molecular Engineering. So our mission, obtaining, maintaining, and training is pretty much what all cores do. Obtain high-end equipment, maintain the equipment and the expertise, and then train people in the collection and analysis responsible and reproducible collection and analysis. So this is us by the numbers. As Kay said, I've been in the Core about 20 years. I started in 2006. I had been a user of the Core in my graduate work at the University of Chicago. And as I was wrapping up, they opened up a position for a 4th member of their team. And so I applied for the position. And based on my teaching experience, I took the position and decided maybe it would be a nice place to work for a couple of years and then go on and do something else. And then fast forward 17 years later, our founding director, Vitas Bendokas, retired, and I was asked to take over as director of the facility. And I was able to hire 2 more staff members. So Khalil Rodriguez and Lorraine Horwitz are our 2 new imaging specialists. We have 14 user-run systems and 3 full-service whole-slide scanners, about 450 active users, from 152 different labs. And on average, those users run about 675 microscope hours and about 1,000 slides a month. So unfortunately for the light sheet panel, light sheet is only about 3% of our total usage. They're not heavily used systems compared to things like the traditional point scan and confocals, spinning disc confocals that people think about. So this is sort of the breakdown of our systems. Point scan and confocal, we are heavily Leica-based. Spinning disc confocal, we have Sora and we have Marianas from 3i. And then we have a live DSU system from Olympus, Evident. And then we have our 4 wide field systems include a stereo microscope and a dedicated histology microscope. And then, like I said, we have our 3 whole site scanners for the slide scanner service. So we currently have 2 light sheets in the facility, but at one point we had 3. So I'm going to talk about all 3 of those different light sheets. We have the 3i Bessel beam light sheet, which is essentially a clone of Eric Betzig's original lattice light sheet design. We had the Zeiss Z1 system, and then it didn't die. We gave it away. It went to live on a farm. And then we also have the Miltenyi LaVision ultramicroscope too. And you'll notice that it doesn't have the beautiful teal and orange body that Miltenyi now has on it. This is the first generation of that system. We acquired it right around the same time that Miltenyi took over the LaVision company. So this is first gen. So some of the challenges that I'll talk about with this system are purely because this is first gen. Some of them have been solved. So usually when users come to us wanting to do a light sheet project or a project that we think could benefit from light sheet microscopy, we start the decision-making process breaking down by sample size. So in our lattice light sheet, these samples are on the smaller end of the scale. Single cells, cell monolayers, anything up to small organoids, maybe a blastocyst. I'll talk about all of these in a little bit more detail in the next slides. So we're talking about, things up to about 50 cubic microns. And then in our Z1 system, this is our embryo system. So larger organoids like brain organoids, Drosophila embryos, zebrafish embryos. We've even had octopus embryos in there. And then the Miltenyi system is for large cleared samples. So this is not a live sample system. This is purely cleared samples. Our system goes up to a cubic centimeter. There are now systems that can do whole adult mice. So some of the image processing challenges that come with those larger samples have become even greater with newer models, but we still have some issues at a cubic centimeter. So, and that's a set of whole adult mouse lungs that we've imaged on our system. So the lattice light sheet has that classic Harry-Schroff inverted SPIM configuration, where the excitation and collection objectives are at right angles to each other, coming down from the top onto the sample holder that's down here at the bottom. And one of the challenges that we face with this system is that the sample has to fit on a 5mm cover slip that goes into a small metal holder that is then bolted onto the piezo in a heated bath and then lifted into the objective configuration. So that's a little intimidating for some of our users. This we purchased as a beta. We were offered the beta system from 3i in 2013, and we purchased it with funds collected from various departments around the university. This is actually one of our more successful experiments that we've run in this system. It's a 50 micron cubic volume, 1.54 seconds per volume for almost 13 minutes. And so you can see how your data collection starts to kind of explode and expand. But this was actually a collaboration between 2 different graduate students. So Jill Rosenberg and Daniel Cow both worked on this project. Jill did all the wet lab side, everything up through image collection, and then she passed the data off to Daniel, who created an image processing and data analysis pipeline to track the T-cell receptor movement when these green T-cells interacted with the red antigen presenting cells. That is not what I wanted to do. Okay. So this is our Zeiss Light Sheet Z1. It's got the classic Stelzer orientation, a bit like the movie that Kay was presenting. It was purchased by a PI at the university with the departmental funds from their department, and then they agreed to add it to the core. This zebrafish embryo was one of our more successful samples, done by Anna Beiringer and Vicki Prince's lab. Some samples that we have not had success with in this system, we can't even go to a cubic centimeter in this holder. So organoids tend to be too small. And then the particular model of Z1 that we had was live sample only. So we were only able to image in aqueous media. We could not do cleared tissue. There was quite an adventure with some cleared mouse hearts and some corn syrup that maybe we could talk about over coffee, but it was at that point that we found out that we could not do cleared samples, and then we talked to Zeiss, and they basically said, yeah, you can send your entire rig back to Germany, we will completely redesign the light path, and then you could do cleared samples, but then you won't be able to do live samples. So the Z1 was not, like Kay's movie, compatible between both live and fixed cleared samples. And then in our Miltenyi Levision ultramicroscope, this is one of our more successful projects here from Frank Coe at Rush University. And this is one of the more impressive clearing protocols that I've ever seen. He managed to keep native TD tomato fluorescence with a Pegasus bone decalcification and clearing process. He's since modified his protocol from his original paper, and now it's more like an iDisco with decalcification protocol, but he still keeps native TD tomato fluorescence. So our most successful samples there are iDisco cleared samples. We have also done some of the sucrose-based clearing methods, clarity, scale, and then, we're trying, I don't even remember what it's called now. There's a new rapid one. But we've managed to do those. Unsuccessful samples, so the smallest sample that we've managed to image in there is an adult mouse lymph node. Anything much smaller than about 7-8 millimeters tall is too small. The halo configuration, basically your sample sits down on a tiny little stage inside the cuvette, and anything that gets too short, you get shadows literally from the Velcro spikes that we're trying to use to hold the sample on the stage with gravity. There are now new holder mechanisms in this type of light sheet that do a much better job of holding the sample in place and making high throughput and clear images possible. All right, so here's all the people that actually did the work. I mostly just teach them how to do it and then let them go on their own. And if anyone's interested in any of the publications whose data that I showed, they're all up here. And with that, I'll turn it over to Christina.
Okay, hi everyone. It's great to be here. It's been a very productive conference so far because we've been talking a lot about light sheets, so I feel already I got a lot out of it. So as Kay said, my background, I'm a biologist by training and a microscopist by trade. I got my PhD in my undergrad in Athens, University of Athens, Greece. And then I got my PhD in the University of Colorado. That's where I started working with imaging, but I started working with light sheets when I joined Zeiss. I did a short postdoc at Rockefeller. I joined Zeiss right about the time they were coming up with the Z1. The Z1 was like a year out in the market. And it was about when they were redesigning things for the second version of Z1. And then I went to the Rockefeller, the core, the BRC, where they had the ultra microscope that Christine mentioned, that the very early version of the Milton, back then it was LaVision light sheet. And now I moved 2 years, not even, a year and a half ago, a year and a few months ago, I moved to MSK to the Advanced Light Microscopy Innovation Lab, where we have 4 Bruker systems. I'll talk about that in a second, but before that, I'll give you a little bit of background about the Advanced Light Microscopy Innovation Lab, which is a mouthful, so I'm just going to call it ALMIL. It's slightly different than traditional core. The idea, and I thought it was unique to MSK, but I found out later there's other innovation labs in other institutes. The idea is that we are at the interface between the standard, confocal, the instrument that can be operated in an imaging facility without issues, and the home-built system. So basically, we're in between the cutting-edge microscopy that it's still hard to implement in a multi-user setup. And the idea is that we work with the users that they want to do these high-risk projects. We have flexible and lower cost recovery expectations, so we don't mind spending a lot of time troubleshooting. And then once we get everything good to go with the instrument, or we have standard SOPs, the idea is that this technology can be transferred to a core, and we are there to assess whether that's the time to happen. So in the facility right now, in the lab right now, I have a STED MinFlux, which is a combination system. It's one of the 2 worldwide, actually. It's one in NIH and one in MSK that they have this specific configuration. We have 4 light sheets, which is the LCS from Bruker, the large clear sample, 2 movie beams and 1 true live. We have Sora, which is here. Is it playing? It's not playing. Oh, it's playing. And we have also a 2-photon microscope with FLIM from Leica, the Falcon. So all those 7 instruments are being available to all the users in TRI-I, which is MSK, Cornell, and Rockefeller University. And it's currently the facility is just me. It's a one-woman show. We have 50 users that they're quite unevenly spread between the 7 instruments. As Christine pointed out, the light sheets get like less traction than the Sora and the 2-photon that they're booked probably like 24/7. The light sheet instruments, my idea why this is happening, because again, I had my fair share of exposure to the light sheet technology over the past decade. I feel that still to this day, the light sheet instruments are still considered more exotic. The user is used to the standard configuration when you throw your slide on, you focus, you defocus. And most of my users, and depends on the institute, of course, because, for example, Rockefeller across the street, when all the clearing revolution was happening in 2014, 2016, Kay was there for that too. They don't consider, obviously, light sheet exotic because they've been exposed more to it. At MSK, I find it's a lot harder to convince people to clear their samples and throw them on the light sheet, even though you tell them what the benefits are going to be. So I would say that the challenges that you'll face if you try to implement light sheet for the first time in your institute or facility would be the activation energy is a lot higher than any other instrument. The clearing protocols are lengthy. They take time. And people are like, oh, it's too long. I mean, you're waiting, but still, it's like it's hard to convince them to go through the process. There's a lot of hand-holding in the beginning, from sample prep to imaging to image analysis. We had a very interesting exposure earlier, how big that data is. It's not small, and people come with a flash drive and it's like, transfer my data. It's not going to happen if you have 2 terabytes. Not all samples are light sheet ready, and people don't get that. How many times I had to tell no to people because they bring me a slide and they say, can we put that on the light sheet? No, like, where are you going to put it on the light sheet? So they don't understand the configuration. And even for users that they come prepared, that they have read the paper, they know what we're talking about, learning curve for sample prep and the instruments can be pretty steep. As Kay pointed out, the movie SPIM is hard to learn, the LCS also. And the time required for image processing, which we have as a big selling point for the light sheet, we're like, oh, you're going to acquire really fast. The process, the the time required for image processing offsets the image acquisition. It'll take 10 minutes to acquire and like a couple hours to process sometimes. So they have to be prepared for that, because at the end of the day, it might take a little bit of time, like half or 1/4 of the time to acquire your sample, but then the processing will take a couple hours. So at the end of the day, you're still gonna take 3 hours before you have usable data in your hands. So that's stuff that you have to clear up with the users in advance. So this is an example of the true live, the Bruker that we have. This is a pre-implantation embryo. This is for fate mapping experiments from the Hadjiadonakis lab. And you can see for 36 hours, this thing is alive and kicking, and you can fate map like every single cell. This is a beautiful experiment. The person who's using mostly the true live is a postdoc in the Hadjiadonakis lab. For his experiment, that was the only solution, so there was no discussion. For other users that I'm like, okay, but these organoids, you can imagine the true live, they'll tell you, I'm imagining it on the shore, it's good enough. You know what I mean? It's like, it takes time to convince them for the value of switching to a system that they don't understand anything, and you might have to align the lasers in the beginning. The other example, let me see, this one. Okay, so this is, I picked the images based on the cases, because this one was, somebody who studies metastasis with cells through the leptomeningeal tubes in the brain. And when I saw her presentation, I was like, why don't you do that on, don't you clear the brains and image the entire brain? Because there was like just a few cells that she cares about throughout the brain. I was like, oh, we do it with slicing, it's fine. We slice and we like image like serial slices like through the confocal. But I'm like, this one is gonna take you like an hour to image, and you can have your entire brain ready. It took about 6 months of back and forth to convince her to do it. Of course, when she saw it on the LCS, she was sold, but it was a hard sell. She was like, and you can see the glial cells in the leptomeningeal tubes. She can count, now we have like a pipeline on IMARI, she can count them on the entire cell, and the entire brain, not just the slices, but it took a while. This other case is somebody who was great with the clearing. This is a pancreas that is the innervation on a pancreas on a mouse that has pancreatic cancer. And she wants to study how the innervation changes between the healthy and diseased animal. And she did the clearing, but then training her to use the LCS was like 6 very painful sessions. And she still doesn't feel comfortable to use it on her own. So she's like going back to what she was doing. So I feel that's the biggest thing that you have to keep in mind, that the activation energy and like the training might be more work up front with the users. Okay, so just like to tie everything up, how to choose the right light sheet for your facility. And I think in my mind, and based on what we saw already, there's 2 main things that you have to clarify from the beginning. Do you want to do live samples, or you want to do clear samples? The movie spring that we also have and Kay described can do both, but you have to change the octagons, which is a whole part of the microscope that has 4 lenses. That thing is 80 grand. It's not like something small that you switch around. You have to take that off, put the other on, align everything, and make it ready for live imaging. So it's not something that, yeah, it can technically do both live and fixed, but with, you have to do some work up front there too. It's not something you can change midday and image like later the day, like live. And then which one your users need most will determine how you're going to invest your money. And then after you answer this, oh, no, hold on. No, that's correct, yeah. That's correct. So there's a lot of commercially available systems. When I started like working with the light sheets, it was just the Zeiss, the 3i just starting, and the LaVision after that. That was the 3 systems, and now it's like a slew of instruments. Zeiss has 2 light sheets now, has the lattice light sheet, and the second version of the Zeiss, not the one Christine had, but the one after that, right?
Oh, yeah.
There's a new version. Live Canvas has like really cool instruments. It's coming, I promise. The Bruker Luxendo has 5 different instruments. The Papillo is the latest one, which I thought it was a replacement for the True Live, but it looks like it's not. It's kind of complementary. That's how I was told to view that. And the 3i has the lattice light sheet that Christine described, and another one for larger fixed samples. But wait, there's more. the Viventis, which is the only two between the True Live and the Viventis, the only two that they can do pure live samples. The Blaze, which is the granddaughter, I guess, of the ultra microscope, and that's a fantastic instrument currently because you can switch between lenses automatically, and it can generate massive amounts of data of like whole mice, whole clear mouse. This one has like also a bezel beam, the Lighton, which I didn't remember, and this one, which is here, actually Milton is here as well, but the Alpenglow, which I found out a lot about it this morning, we should check out their booth. It's sort of like also an instrument that can do both live and fixed clear samples, so you should definitely go check it out if you haven't already. The Miser Tilt, which was this little device that people, like, you know, the company marketed it as you just put on the stage, it makes your light into like a sheet, and then you image. I've never seen this thing working, so I'm not gonna say anything about it. The alignment was like painful. And the Slice, which is a very small microscope, the cost all included is something like $70,000, and it performs beautifully if you need just 3 lasers, one like for small samples, you can do half a brain easily. If that's your budget and that you want to do only specific things, that works too. So somebody will pay, okay, this is way too many choices, and my applications are complicated. Should I build my own? No. No. Like, every time somebody will try to trick you and tell you it's only going to cost you like that much with the material and the lenses, just say no, walk away. Because it looks like fun, it makes you feel very smart because you're just kind of into like this optics, like, you know, heaven, but it's a time sink and a fun sink. Like, it's not going to get you anywhere. Wait for like your idea to be commercialized. That's my take on it. For a facility, if you're in a lab and that's your research and you're going to have a dedicated person babysitting that instrument, go for it. But for a facility, this is not a good solution. Okay, but what commercial system? Again, there's so many choices. So know your user's tolerance for complexity. That's the thing. Some people are better with going there and aligning the lasers for 20 minutes before they start. Some people want an turnkey system, and those people usually don't do very well with light sheets. That's my experience. Carefully consider the sample holder. That can be tricky too. How big is your sample? Do you want to embed it in agarose? Do you want to clear it, or like, do you want to clear it with a certain method? I agree, the C3D or all those in movie spin, they're a nightmare, because the C3D, for example, as it starts solidifying, the refractive index changes. If your acquisition takes more than 10 minutes, you can visualize the refractive index changes. That's ridiculous. So try to make sure that you have the right clearing method for what you want and the right holder. How does the vendor software will handle the processing, transferring, storage? That's important to know, and that's important to know because you have to talk to your IT beforehand. Don't ask me how I know that. And make sure that you have a plan, because the data can be really big. Like the original, if you do let's say 2 colors on one brain, it could be easily 1 terabyte, and after processing you add a little bit more with the Imaris file, could be down to half a terabyte, but still, this is like the final folder of this experiment, could be a couple terabytes. So you have to know what to do with that data, how to have your users transfer that data, and what's your institutional policy about that. Demo the instrument for as long as you can. I cannot stress that enough. Don't have anyone convince you that, oh, you're going to see it and you're going to buy it. This is a huge commitment in time and funds. Demo it with real samples, keep it for a significant amount of time to be sure that it fits your needs. Because nobody's happy after you purchase something you don't like. The company's not happy because you get consistent complaints, and you're not going to be happy, and your users are not going to be happy. And ensure proximity and availability to support. Things are going to go wrong, I can guarantee that. So it's important to know that your company, your service engineer will be available to come and fix stuff when they need to. All right, so takeaway message. There is not a perfect light sheet for all jobs. You have to find what fits your needs, and it's worth the investment. Talk to the companies, figure out how do they do things, and just kind of be super anal in the very beginning to figure out what are your needs and how you can cover them before you commit financial to something. And this is my favorite light sheet image ever. It's some of you might know Jim Sims is the Hamamatsu specialist, and he has an extensive collection of those amber embedded fossils. And turns out that the amber has a refractive index 1.55. So we put it in a light sheet microscope with some ECI, and we imaged this mosquito, which is like, I don't know, millions of years old. And that's how old your samples can be, and you can still image them. So, so yeah, with that,
So
I
none
think
of
with
that,
the
I have to image this tomorrow nonsense.
no, exactly. Don't.
Does it come wrapped in foil?
All right, well, thank you guys. So that was our perspectives and our overviews. So then from there, we're happy to take
Open it
any
up to
questions. If anybody has any questions or anything. Yes.
I was just gonna kind of comment and reiterate that the comment you made about building a light sheet in a core facility. I had someone come to us to say that the paper and things that was supposed to cost like $4,000 to build a light sheet. They're like, we should build it. And I said, well, we have this Zeiss light sheet that we are trained on, and we're glad to help you. But we want to build this one so that we can practice on it, and we'll test our things on it there, and then we'll come to yours. I'm like, how about you consider spending the $4,000 in our core, and we can help you with it. I actually did convince them.
Hi, really great talks. So we also have a light sheet in our facility. We got it 5 to 6 months ago. It's a live canvas smart spim for clear tissues. And we have gotten great feedback from our users on the learning curve. It has been easier to train somebody on the light sheet than on our confocals. So for us, right now, at least in the last few months, we have realized imaging is actually the easiest part in the whole pipeline. The bottleneck has been on sample prep or data management, which comes after it. So I'm wondering if any of your core facilities provide any sort of support for tissue clearing on that aspect.
Yeah. This is on. Can you hear me? I don't know what's happening. Hello? This way's on. Okay, sorry. Yeah. So yes and no. So that is one of the. So first, my scientist has a theory, and her theory is that if you are good at video games, you will be good at light sheet because you're used to thinking in 3 dimensions. And so it has really been true. Our newest, our undergrads, our first-year graduate students are the ones that pick it up right away because they're used to that whole joystick, it's moving in 3 dimensions, and I'm shooting stuff. I don't know, but they're really good at it. And then the seasoned microscopists are the ones that struggle the most because it's not what they're used to thinking about when they think about imaging. But as far as the clearing, we get that question a lot. Well, will you clear this for us? Or can we just, bring you the samples? And we don't do the clearing for them, but we do have sort of a skeletonized protocol that we give them. And we're like, this is how we do it. This is what we walk through. The other thing that we do in our core is that we limit them. We're like, you bring the sample, it's iDisco. That's what we're gonna do. And we don't really go out into doing having all these different types of samples. And that way we can kind of become experts in that one technique. And then we can offer kind of our advice on that and our experience on that without having to juggle a whole bunch of different kind of ways of handling it. And that's actually worked really well for us as a smaller core.
We don't really provide clearing support at all. The only place that I know of that has a full service clearing protocol is University of Minnesota. They have their whole lab set up, and they have a wonderful clearing fee for service. And I would love to set something like that up where we are. We just don't have the staff time to do it, unfortunately. There are companies that sell ready-made kits, and there are lots of I think it's iDisco.com has all the iDisco protocols, and it's up to date. People keep adding to it.
I think it's .info.
Or it's maybe it's .info. .com sounds, that's probably wrong. .info or .org, something like that. But yeah, like I said, we aren't able to provide any sort of full service clearing. We tend to refer labs back to other successful labs. So Frank is one of my greatest resources I'm like, okay, let's get Frank's protocol. So I have Frank's protocol on file, and I send it to people, and he does not mind whatsoever because now his data is all published, and he's a very helpful person. And in getting users to collaborate with each other has been very helpful at our university.
Because I don't have the bandwidth to help everybody with their clearing, what I do, I have consultation beforehand. I give them the clarifying tissue clearing, the review, to read what this is and why you're doing it. I give them a protocol, same idea as Kay said, like a basic protocol. I tell them that they are free to choose. There's free choice, like what clearing method you want, but
Really
I
wanna
tell
just
them,
go-
I tell them what's going to work best in our systems. I think iDisco now with switching to ECI, but ECI has problems with the chromatic dispersion. It's complicated, but switching to ECI because it's cleaner, you can do it on benchtop without danger. In your case, because that's another comment I wanted to make, because you said training was the easiest part. That's the advantage of the smart spim. It's like so easy to align. It's amazing. It's like two knobs, like you just align it and you go. It has some other shortcomings. I feel like the sample mounting can be tricky if you don't glue it properly and all that. But they have also a whole line of clearing agents, and it's expensive. But if the user wants the easy way out and they're willing to pay, I would do that, and then just kind of fill up the reservoir with their liquid or with oil, and you're good to go. But if you get sucked into that, everybody will want support in the new methods that they just read, and it works so much better, and then it's like, it's never ending. So I think saying,
That's true.
saying like, and sometimes when you want to maintain endogenous fluorescence and you want to avoid clearing with solvents, with organic solvents, I see that, but an extra step of staining with a technique that's been very well established and it's going to give you a lot better results, it might worth considering. Again, at the end, you can lead the horse to water. You cannot make them drink, but I refuse to get involved in that because it can be a never-ending game. So.
That's true. We've had that before. Like, this protocol is so much better. Why can't we use this? I'm like, you go do that. You bring me the evidence that it's so much better, and then we'll talk about it.
And another discussion, the BAB thing, where people would come and be like, I want to image in BAB. No, you won't, because you're going to destroy the optics. But some people insist because that paper that I want to replicate has BAB. I'm like, okay, do it in your lab then.
Or just, image and ethyl cinnamate, which
Yeah, it was the same thing. Yeah.
Hi, I just wanted to talk a little bit about the actual sample mounting, just because that's something that has just been so different for the different samples, and ways to get good optical access without obscuring or destroying the sample, especially as you're changing between the clearing methods. I just figured we'd discuss kind of what's worked for you guys.
yeah, it's really sample dependent, but yeah.
No, you can start this time. I start all the time. It's your turn.
It's fine. All right. Well, so particularly for things like the movie SPIM, the ZIC-1, those embryonic level ones, there's a couple of different ways that you can go. So most of those, the orientation is you've got your 2 illumination objectives, and then you either have a single or a dual collection objective, and the sample hangs or is stood up on a pillar in between, right? In the Z1 configuration, we use either FEP tubing. So FEP has the same refractive index as your agarose that the sample is embedded in. And that's for samples that need sort of that low melt agarose that's a little bit too squishy to be extruded from a capillary, because that's your other prep method, is to do a firmer setting agarose that you pull into a capillary with your samples, and then it's extruded from the capillary and you're just looking through the agarose rather than the glass of the capillary. And then, so those are the 2 main sample prep methods for something like the Z1. We have been able to get creative in the past. For the octopus embryo project, an octopus egg is sort of teardrop shaped. I don't know if anyone's ever seen an octopus egg, but they're sort of teardrop shaped. And the embryo hangs out down at the bottom, and the rest of it is all yolk. And so we couldn't hang the teardrop into the system because the embryo wouldn't have the correct orientation for the objective. So what we ended up doing, we had a holder that would hold a 1 cc syringe. And so we put that in, and then we took It's actually my mentor, Vitas Spindokas, who engineered this whole system. He took an acetate strip and made a little bit of a loop and attached that to the tuberculin, so that was always hanging into the chamber. And then Carrie, the graduate student who did the project, would take her egg and slide it horizontally onto the acetate loop with the part with the embryo poking out the end right in front of the objective. So she had a face-on view of her embryo, and the sheets were able to illuminate the embryo outside of the acetate strip. So you can get creative even with some of these more constrained mounting situations. And then I know in the movie, they have like a pillar system, so it's a little bit different still.
And I think also for the live samples, there's definitely like, there's always like, well, can we use this? Can we use that? For ours, the samples are generally so small in the live sample that we are kind of limited to what can fit in the FEP tubing, because even if you can get something in there, you don't really have the range of motion of the stage. But for the cleared samples, what we've done a lot of, and what we found has like changed everything for us really, is that if you embed the sample first before you clear it in agarose, and that agarose can be square or it can be a pillar. Like one thing we do a lot of is like a 1 ml syringe filled with agarose, and then you can put your organoids in there. You can stack like 3 or 4 of them up, and then you have like a perfect pillar. We had this one group that wanted to image a hind limb, and so it's like a triangle, and you're trying to hold, the way that that holder works is it's like a clamp holder, and so you bring the top down, you're trying to hold a triangle in like a top and bottom clamp. But if you just mount that in like a little square of agarose, then you have a lovely little cube, and that's super easy to hold, right? And then you also have a lot of buffer reason, so you're not compressing your sample because you have all this agarose around it. And when we kind of realized, when we kind of like really just started using that a lot, like relying on it a lot, and like just getting the right agarose, and it clears so well most of the time. It makes it a lot easier, and especially in that kind of hold it like this, and that mechanism, and it really has made the biggest difference in the world. Like things that would take like out, like there was a point I was like, I had a cochlea, I'm trying to glue it to a toothpick without like making, because the glue makes it not clear anymore, and like I spent like hours, and then we were like, oh, we just dropped these in agarose, and it's trivial, like, to mount it at that point. And so it really just, sometimes just thinking around the problem in a different way. It can make a difference.
For the larger samples, because there's going to be people who refuse to embed in agarose, although all samples can be benefited from embedding in agarose, even if it's a thin layer, because you have a buffer zone for your sheet to go in and not get diffracted before it hits your sample. So you're going to see like straighter, like better imaging anyways. But for the people who don't want to do that, super glue is your friend. Find like a super glue that dries fast, like dry a little bit the sample, like for a brain, for example, for those holders. The holders from Bruker are also a work in progress. There's a whole thing. You're gonna 3D print a lot. That's the other thing. You can design and 3D print a lot and find out what works for you. And there's tricks to do that. Like feel free to contact me. Like
yeah, no, that's one thing we did. Like you get the regular one, and then you just tweak it.
Yeah, you tweak exactly. You get the original design, and then you play around and you make it fit your sample. That's
Yeah. Does that sort of answer your question?
Yeah, no, I mean, I think we're actually, we'll want to chat in more detail. It's been a struggle for us, but.
Yeah, I mean, the problem is there's a lot of different configurations across all of the different light sheets. So it really depends on your specific use cases, like what kinds of things are possible.
And I think that's one of the other things when you're looking at a system, the sales rep, like, it's only this, like, it's just this, like, the alignment's just these 3 knobs, or to mount the sample, it's just to put it in this clamp, or it's just to set it on this pillar, and it's like, just becomes very different in a core facility when you don't have just one type of sample, right? I'm not just gonna be imaging a half a mouse brain every day for the rest of my life. Like, I'm gonna have somebody come in with some hand, with like, they wanna image the middle finger of a mouse paw, and like, how am I supposed to do that? Like, and it's a whole, it's like a whole thing, and it's not like you just can do the same thing every day, and that does make it a little bit more fun, but a little bit more challenging too.
one more comment on that. Some companies, like the companies that they've launched a newer product, they're more open to discussing. And if you find the right people and talk, they might help you in designing, or they're gonna design for you. If you go to Zeiss and say, I cannot embed my sample, they're gonna be like, yeah, it sounds like a you problem. But like, but if you find a company that's like willing to improve their product, they might be also willing to work with you more and figure out solutions for your specific problem.
I think that's a good point too, because like you'll meet lots of companies where like it's all physicists, and then that's great for your optics, but then they don't have necessarily the experience to be like, oh, this is how I used to clear samples when I was a postdoc, and that can be really helpful to that relationship, and just finding a company that kind of fits with what you need to have help with and what you need to kind of build on in your facility. I think that can make a big difference too, and that's going to be different for every facility as well. Like, yeah, no, for sure. Do you guys have any questions?
For each other? I mean, we've been talking about it.
I don't know. I'm just saying. I just don't want to exclude anybody.
Well,
What's up?
I think we need to go back and forth because she's big data processing.
I know. No, we're going to
We have
kidnap her
questions for
later.
you. Yeah.
We can have our own little round table.
Yes. The full pipeline.
So all your facilities are pretty well established. I'm just curious,
No.
if you were to set up a new light sheet facility today, would you do anything differently?
Oh, that's a good question.
Oh, I would definitely get newer stuff. I mean, so everything that we have is first generation stuff, right? And as much as I like it and as well as it has worked, right? We do have publications from all of our equipment. That the dialogue that we have had with companies and the things that we have told them that are stumbling blocks for our users, you can see the progression in their instrumentation and the improvements that they've made across generations based on feedback like ours. So, you know, being an early adopter is fabulous for some reasons, but it's not so fabulous in other ways, right? So if you can wait until a technology is a little more firmly established, as it's clearly becoming, right? It's still considered exotic, but I think it's developed quite a bit since like 2013 when we took in our first light sheet. I think that's a very powerful thing, just to kind of, you know, don't feel like you're missing out by sitting back and waiting for things to become more established. You are actually waiting for things to become more established and to become more turnkey and easier for your user base.
Yeah.
So we still use ours on a limited. I have a handful of labs that still use it. I know, I don't know if anyone from St. Jude's is here. I know people from St. Jude's are here. I know they use theirs, they love theirs. But you would be surprised at the number of phone calls that I have gotten of, I'm a new technical director in this core and we have a 3i lattice that is sitting and collecting dust. And it's, yes, that is very much what has happened. And I will say with ours, it was about to become end of life, and I had a very frank conversation with the head of our heaviest user lab, and I said, look, the laser launch is gonna die, the cameras are getting old, you know, this, that, and the other. If we don't invest money in this system, it will be dead within a year, 18 months. And fortunately, he was able to give me the money that I needed to refresh the laser launch, to put a dual cam system on there, and to keep it running for himself, but he was generous enough to give that equipment and keep it in the core and keep it running for everyone. And not everyone has that level of, you know, patronage essentially for their systems. So yes, a lot of them are collecting dust, unfortunately. But again, based on our feedback, they have refined. So if you buy a 3i vessel beam light sheet now, it'll be a more refined product than it was when we got it.
I would say too that for us, when I first got the movie SPIM, the first 2 years I was like, oh my gosh, I made the wrong decision. I was like, this is terrible. It takes 3 days to line it, and it's you know, it's terrible. And I was, but by now, like I said, it's a pretty good system. Like, it does everything we needed to do. It only takes us like, you know, everything is working now. Like it really just took us a while to learn it because it was so different. And so I think that all of the things that I was hoping to be able to do with it, I actually can do with it. And it's not as bad as it seems some days. When you turn the wrong screw and you have to spend 2 days realigning it, yes, that seems terrible at that moment. But overall, one thing that we've been able to take all comers, like we have that sort of flexibility, and that does come with a price. But now that we can switch to Octagon, we can have it set up in 2 hours, and that's a big difference. And getting to that point was hard, but I was like, oh my gosh, I made the wrong decision, and I don't know what I'm gonna do. But we got kind of past that. But so I think it can also change day to day if you do something different or not. But yeah, so I think.
So in my case, because Kay had to write an S10, and it was a very deliberate decision. You got funded, and you bought that instrument. I walked into a situation because the Alamo, I didn't mention that, I guess, but it was established when I was recruited. That was it. That was the beginning of me and the lab in the MSK, and the 4 systems the light systems that were already there. They were pre-purchased by somebody else. They were been sitting there for 3 years. So, and they haven't been heavily used in that time, no, probably 4 years. So the instruments I bought was a Minflex, and I was there for the beginning of it. So I consider more my choice. The Brooker instruments, as Kay said, they're not bad or anything, but they're they give you more degrees of freedom and more responsibility to align them properly. And it's more stressful for me as the sole manager of the facility because the problem is that when you acquire an image on the LCS, for example, right, and say you screwed it up and you have misaligned the lasers and you get doublets everywhere, and the user doesn't see that, takes the raw data and is like, oh, I'll process it later, and walks away, then you cannot fix that. Because you have dual illumination, but you detect with the same camera, so that's unfixable. You have to reshoot the whole image. So you have to train the users well to make sure that they look at the samples, make sure that they don't do anything screwy in the beginning. And that creates extra stress for me. I would probably go with a smart spin, as a beginning instrument for larger samples to see and also most of the people are the researchers are physicians that they want to see large samples, they want to see metastatic tissue, whatever. We're not doing this one person that does live, but that's not. So I would probably make different choices in the instruments. But as Kay said, the instruments per se, they can do what you want them to do if you're willing to invest the time.
That's the other thing. We were running an S10. We had lots of time. We were kind of established pretty well as a resource, not a big one, but we were established. And I could say, okay, I'm bringing this technology on, but I need another half a person.
Yeah.
I'm going to have to hire somebody if we want to do this. And having the flexibility and the support from my administration to say that makes a big difference because, again, at our institution level, there's most of our research is graduate student run. There's not really going to be a time when they can just come in and align it and change octagons. That's never going to happen at my institution. So to be able to say, I need a person to do this, that makes a big difference. If it was okay, no, then I would have had to go with a different system. I'd be like, well, then we can't do live. We're going to have to pick something that's simpler so that because we need that support to do it. So I think that's another big point. What are the other factors? It's not, your users think they're the only ones that matter, but other things matter too. That you have to kind of make sure it's going to work out.
And the other point, to your point about the data, is I walked into a situation where they bought the Hive. The Hive is just kind of basically a big, server from Luxendo that, you can connect to your instruments. But they didn't talk to the IT people, and the IT people walked in with attitude, being like, "What is this doing here?" I'm like, "I don't know, I just got here." So that was those discussions have to happen earlier, and I didn't have this, so I would change that, because again, the Hive, is it useful? Probably, but not the way it's set up right now. So there's things like that, and maybe do more of legwork, because I guess when you got your system, you advertised it and you said, well, we're having this new technology coming here, whatever. The technology was already there, and some people were already bitter about it. They're like, oh, it doesn't work as it should. And I had to fight that it gets uphill battle, be like, no, it works, and it can do good stuff, but yeah.
Yeah, so, hey, Steve Pemberton with Alpha Biosciences.
Hi.
We're one of the manufacturers of light sheet instrumentation, so thanks for mentioning us. So the question I have is from a vendor perspective, how can we help support this community? You have put up that pie graph where light sheet was unfortunately just a very, very tiny sliver. And that's one thing, I've been with the company for almost 4 years now, and it's a very, very lonely voice out there in the wilderness pushing 3D spatial biology. So one thing I want to know is how can we work better with you to help your users adopt this technology?
Of course.
So one of the things with the Chicago system is it's exotic. There are 16 lattices in the world.
It is exotic, exactly. It's a little bit exotic, yeah.
Right? So there's a significant learning curve to drive lattice. Even as a builder, right? It's tough.
Yeah.
But I noticed that on your deck too, and that was alarming to me. But Steve's question is still very important for us commercially. How can we support you? Because throughout this meeting, whether it's this panel or another panel, I've been thinking, you guys are one step away from asking a commercial guy to solve all of your problems. I'm serious. Really, honestly.
Can you do it?
Yeah. I think you can.
Thank you.
So I think it's we do a lot of outreach, right? And there's tissue clearing webinars that are on our website that you can have access to, and common tips and tricks of clearing and artifacts that you see and all this stuff. Do we need to do more of that?
So I have thoughts. So
I know you have that. You go first. You go first with your thoughts.
because I've been working on these two ladies like all day long. I'm like, we should have a course. No, I feel like that's my light sheet course. No, that I feel like
I already do MSJ courses. I don't need to do it.
no, you listen to me now. So no, my idea was that I've been to courses, light sheet courses, a few actually, and the Southwestern one when it was just builders like Kevin Dean and Fioga, and that's cool stuff, but that's way far removed from what we need as facilities. And even the course in MDI was great, but again, it was great to see all the new technologies, but it's it's a week of just looking at the new technologies. There's no training of the users happening, and I feel that would be useful if there were more events sponsored like that, like actively explaining, explaining why you cannot put basic,
What
but
she
not
means
basic.
is, will you sponsor her new
Of
course?
course, more hands-on.
Yeah.
kinds of things.
No, I mean, if you explain why you cannot put a slide on a light sheet and why this is limiting, I mean, it's basic, but not too basic for a biologist I think about microscopes day in, day out. I love this subject, but the user who comes in my facility thinks of their cells and their embryos 24/7, and then they just this one moment that they need a picture for their paper, they're just gonna come to me and ask for that. So I cannot ask them to do the legwork. Yeah, I'll send them the review, but I feel it's on me to try to train them and understand what the clearing does, why the lenses are the way they are, and stuff like that. And we can do that. I mean,
The other thing, though, I'll say from what you said before, I think that there's a lot of rhetoric that's just not helpful. That it's so easy that you can just do this, that you can just do that, and then a user comes in for an instrument that isn't just do this and isn't just do that, and they're like, this doesn't work, and they won't come back. And you only get that one chance.
Yeah.
Like, you convince them for 6 months, they've spent all this time clearing and staining, and then they come in and they walk away with trash, you're not gonna get that person back. And I think that's one of the biggest things is these instruments, especially what we've struggled with, is the instruments are still kind of in the learning phase, and you have to have the users that go through that, but you lose half the users when it doesn't work. And I think that this idea that it's quick and easy, it doesn't actually get more people. I think it discourages people when it doesn't work perfectly right away. And I think that's one thing that we have fought against. It's like, oh, but this paper saw this, and then I'm like, okay, let's dive into that paper. They processed the hell out of this image, and they put it out there as a light sheet image that they took, and there's 4 filters in this, and then a segmentation. You know what I mean? So there is this kind of beauty, simplicity of it, and that's true to a certain extent, but at the same time, we have to be realistic and transparent about what goes into that. And I think that is a real struggle that I have.
So I wanted to address your statement, Christina. So there's 2 really good courses. The LSFM course run by Gary Levsky. That's going to be in Dresden course, Emmanuel, Renaud, and Pablo. I think those are the best courses, and I don't know if people know about them, but as far as getting hands-on a rig and twisting the knobs, and then also getting the feedback on tissue clearing, we send the best people there. We send our A teams. Court goes there all the time. I mean,
That's
it's
one
right?
course a year.
Exactly.
You know, that's what I mean. We can expand that, and that's what I was thinking. Like, it doesn't have to be an extensive, like, full week course, like, Cold Spring Harbor, or like, somewhere,
Right.
Just move everybody. It can be on the institute, a 2-day workshop, hands-on. And again, I'm sure I'm gonna have like institutional support, but also need support from like the people who are gonna bring an instrument, and like on top of my instruments, we can see an extra light sheet, stuff like that, like just make it more accessible. Also, with the current financial climate, you cannot ask people to pay $3,000 to send their student to Bar Harbor
Or
for
to
like a week,
Dresden, yeah.
or Dresden.
Well,
So
the thing from a manufacturer's perspective is, I ship an instrument somewhere for a
It's
week
expensive.
and install it and line it and ship it back out. I mean, that's a big lift. What we started doing that we've had some success with, and we're going to accelerate a little bit more this year, is virtual tissue clearing seminars, where people can jump into the process based on a protocol, and they can get some help and support from us through Zoom and virtual support along every step of the way, and then send us tissue to image, then we'll grade it and let them know if they have surface staining, do they have depth penetration issues, do they have pore clarity, and give them that feedback. Now, it takes a while, but I don't know anything like sheet tissue clearing, it doesn't take
Right.
Well, exactly, and I mean, to your point, right, all of imaging is an iterative process, right? Your staining informs your imaging, informs your image processing, informs your staining. And the biggest hang-up a lot of people have with light sheet is that timeline is expanded considerably, particularly when you're talking about cleared sample imaging, right? Your sample prep takes 3 or 4 times longer than it would for a confocal sample. Your actual imaging is the easiest part, as you were saying, right? And then the image processing, the whole cleared mouse lung set that I showed, when we denoised that, it took 12 hours, right? People are not used to a 12-hour denoising step. And that's a real problem for a lot of our users because their PIs are thinking this is on the scale of confocal. That's the timelines that you're looking at. And it's really not, it's not at all that sort of scale. Frank spent 9 months perfecting his Pegasus bone clearing protocol. And because Frank is a postdoc and he has that kind of time to do that. Your average graduate student is probably not going to invest that kind of time. So those are some, and companies can do all they can to speed things up, but you also have to understand the sort of mindset that you're walking into.
The other thing is that most labs have years, decades of optimization in their imaging protocols, and they don't really think about that. Like, you've been doing confocal for 12 years, and that's been optimized in your lab that whole time, and now you're the first person doing this. That's like inventing a new wheel. Like, there is a whole thing. I want to give, Michelle, did you have, I don't want to keep you from talking.
It's slightly related. So as we've tried to support our light sheet imaging projects, we've run into all of these phenotypes of users, but the one that we consistently get asked by administration to help serve is the ones who say they just want a flyby, they're not gonna analyze it, I just want a beautiful movie to put in my talk. And our philosophy before had been like, no, we want to train you, we want to get quantitative data, but like with small staff, right, we calculated that out and I was like, we can offer you this, right? It will require this many consultations, it will require this much staff time, we're not going to teach you how to use it, you will be there while we acquire, and we will deliver this. And everybody who wanted that was like, that's way too expensive, right?
Yeah.
And so I'm wondering if anybody has threaded that needle where you can still meet that need, but still recognize the expertise it takes to get that just image. because we haven't quite found that yet, and I know that that's been out there in the field.
Yeah, we basically do like the first session is just like, let's see what we can get. And we go through that troubleshooting process, usually with us running the instrument. So like while we're still working out, like is the staining, is the clearing working? We don't really hand it off to them until we get to the point where the imaging is working. Because what's the worst is when you train somebody and then they don't ever come back, and that's just such a waste. But again, we're not full cost recovery. If we were, I don't think we could have that flexibility either to be like, oh, well, we're just going to actually waste 12 hours with the hope that you're going to image later, right? But yeah, they need that first take-home image to get them to say it's worth it, right? And so if you're like, no, you have to be trained first, then that even makes it even longer before they see anything that's real. And I think that can, you're right, it's really hard. And I've had that too, like when you do the clearing for us, and I'm like, you can't afford it. Like you can't afford to pay me to do the clearing for you. And like, you know what I mean? Like
So for that 12 hours, right, like how much are you charging to the user?
usually just for instrument time,
Wow, okay.
which is crazy.
Yeah.
No, like, cause I'm saying it's insane.
Yeah,
It's insane. There's no way that it would be sustainable if I had to recover my costs.
Yeah, that's the other point with that the flexibility of recovery right now. I don't know how long it's going to be like that. Helps me attract the user and sit with them and show them what I can do, because that is like valuable time, my time, the instrument time, like the time to process their stuff, to send it to them, to explain. Like this is not sustainable in actual core, which. Again, and also I don't mind being the negative Nelly and tell them that, like, because I used to be the, you can acquire stuff so fast, and it's like, and I'm like, no, listen, it's gonna be hard, it's gonna be painful, but it's worth it. And if, I've just like vendors who were like, oh, but it's only 20 minutes to acquire that. I'm like, because 20 minutes, and they're gonna spend 3 hours like processing it, and they're gonna be pissed at me because they don't have their data in 20 minutes. So you prepare them for the amount of time you're gonna spend, and if you can afford letting them off the hook for some of the time to just see the value, but yeah, it's hard. It's not sustainable.
really hard.
No, yeah.
And they're super proud and they're super excited when it works, but
Yeah.
you have to get there, and that's not easy. And I think that's really hard to teach like administration. Like we have to waste money to make any money.
Yeah. It's marketing, you know.
Well, thank you. So in my facility, I'm also a single like staff member of a core facility. We don't have a light sheet, but we've considered getting one based on kind of our users' need and whatnot. People were talking about what can vendors do in terms of like roadblocks to getting labs interested in this. One thing I've been very interested in is kind of ways to kind of speed up the sample prep process, in particular like active clearing methods. I don't know if anyone here has any of those, or if they had any experience with it in terms of how well that works for them. But also partially to people's questions about what can vendors do. That's a pretty big ask, right? Saying, hey, vendors, help create this new technology. It's very difficult, but in my opinion, as you know, thinking about what do my users need, if we could find some way to remove that, kind of the iterative process could be faster.
I think I've got Adapt 3D waiting when I get home. That's the newest. Well, and that's the thing. That's the newest rapid clearing method, right? There's a new paper every month that's like, this is the rapid clearing method that is going to solve everybody's problems. And in some ways, it does and doesn't exist, right? It's very sample dependent. And sometimes the biggest hurdle in the process is finding the right rapid clearing method for you.
Yeah, and I was specifically asking about active staining methods, actually. I know there's one or two out there that are commercially available. Does anyone have any, has it worked well for you, not worked well for you, or thoughts?
I'm sorry, what did you say?
I find the opposite. I find that.
Well, because you're a vendor.
I think the active clearing, and I come from a company that like espouses, right? But I'm pro passive clearing. But then on the 3D IHC side, we've solved that problem. I think we've solved it very well. But yeah, I mean, I don't know if there's another person here from my company that's going to kill me later. I mean, just when you think about running electricity through an organ, that's just like,
Well, I mean, yeah, we had an active standing ring very early on in the process, and it also went to live on a farm. It went to live in a circular farm in the back of somebody's lab, and it does. You are. It's gel electrophoresis trying to shove antibodies into a tissue, and again, it's very sample dependent. It's very antibody dependent. It's still an iterative process, right? It's still not going to solve all of your issues. It still takes troubleshooting and work. So yes, there are commercial systems that are available that will do it for you and somewhat speed the process up, but there's this perception that speeding things up means that there is less troubleshooting, and that is definitely not true.
I have another comment on that. Doesn't directly answer your question, but since you're one person and you have limited time, make sure that that initial consultation you ask your users, because somebody will come in and it's like, I want to clear the entire mouse. And you're like, okay, cool, let's find a way to do it fast. No, ask them why. Because the thing is like, oh, but I want to see the olfactory bulb. But it would be cool to see it on the and that happens more often than so much. Like they're like, oh, you want to see the entire brain. Why? Like your protein of interest is in the hippocampus. Just like cut that part. Because also like, yeah, it's beautiful to see and make that entire brain for the cover of Cell when you get the paper, by all means. But initially, start with a thick slice, clear that, test your antibodies, because I feel that's the other thing.
Yeah.
People don't do that, and pisses me off. They come in and they're like, I waited like 3 weeks and now my antibody didn't work. Yeah, if you had done a thick slice, that would have taken like a day or two, and you would know that your antibody didn't work, and then you wouldn't try that antibody on the entire brain. So ask questions, because sometimes chopping down the brain or the specific organ to the area that they care about, that's going to speed things up, and you don't need a new method. That's going to make it a lot faster to begin with. And I think, I don't know, you guys experience, that happens all the time. They want the whole organ for whatever reason.
For whatever reason. You know what I do?
Sometimes more
I
is
set
just
up the
more.
scan. I say, okay, you want the whole brain at 20x? Let me do that. I'm like, so that's going to be 18 terabytes and take 46 hours. And they're like, oh, yeah, no, I just need the hippocampus. You're like, that's what I thought. Yeah, but I think that's like that is a part of it too. It's just like, what do you actually need? And then like for the clearing stuff, I mean, come on, it's the cost of science. And I think that is another thing. It's just like everybody wants to do like an automated this and an automated that, and I just think, honestly, it's not that much work. Like you're just like putting stuff in the tubes and then you just leave it alone, and then you come back later. It's kind of, I mean, I'm not offering to do it for you, obviously. So there's obviously some work to it, but I also think that like
solvents, the protocols are so optimized right now. Like, you know, you have to try and screw it up. Like, just do it. And again, like, you know, do a smaller part, like, you know, and then it's gonna take maybe like a few days. I feel like, again, you have to establish a culture, which again, I'm failing, so I cannot give advice on that. But you have to establish a culture where people are just kind of understanding what the limitations are. I cannot defy physics. I can help you, give you advice, but I cannot defy physics. However time the liquid needs to get in there, it's going to take that amount of time.
right. And just listen to me. Like if I tell you to put it in a new tube, put it in a new tube, right? Like just do it. Like, is it really worth wasting like four months of work because you didn't want to use an extra tube? Like there's just like so many weird things like that you just kind of have to, they have to do it, and then you have to be like,
Some
oh,
people have to learn that
that's
lesson for
why it didn't
themselves.
work, yeah. And so,
Unfortunately.
You have to
which
be
is,
aggressive.
again, difficult, because then of course they don't want to pay for something they didn't see. So.
Oh yeah, it didn't work. I don't want to pay.
That was
That's
an
a
earlier
different
one,
panel.
yeah.
That's not our panel.
Well, thank you all.
Thank you so much.
You can always come find us if you have any other questions.
Yeah, feel free to reach out.
Oh gosh, we're going over time.
We held you guys hostage for like a really long time. Sorry.
We're sorry.
We went over time.
It's like almost.
Like 10 minutes.
We're that interesting.
Okay, or that chatty. One or the other.